Welcome to AB Biology 3.6 Cellular Respiration. This is Dr. Joseph. In this chapter, we are going to study cellular respiration, essentially how the energy that is stored in glucose molecules is converted into ATP, which is the energy currency of the cell. And that involves three processes: glycolysis, Krebs cycle, and electron transport chain, together called as cellular respiration. We are going to see how energy flows across the cellular respiration process and the importance of oxygen in that process and what happens in the absence of oxygen. So we looked at photosynthesis before. Photosynthesis is where plant cells have chloroplasts, and the chloroplasts are able to trap the energy of the sun using pigments, and that energy is converted into sugars. Now the energy that is stored in sugars has to be broken down if it has to be of any value to the cell, and that is where cellular respiration comes in. Plants need energy and they are storing up the energy from the sun in the form of sugars, but animals that eat the plants and those that eat the animals that eat the plants are able to use that energy stored in carbohydrates as well. So what happens in cellular respiration is that the sugars are broken down, and most of this happens in the mitochondria. So there are steps outside the mitochondria but steps inside the mitochondria as well, and that causes the breakdown of the sugars, releasing energy which is stored as ATP. So from energy of the sun to energy of molecules, chemical molecules in ATP. That's the bigger flow of energy in the living systems. So let's look at cellular respiration. Cellular respiration involves three steps. The first one happens in the cytoplasm and it's called glycolysis, and the next two steps happen in the mitochondria. They are called the Krebs cycle and the electron transport chain. There is also an intermediate step between glycolysis and Krebs cycle called as pyruvate oxidation that also happens within the mitochondria. Okay, so just to take a quick look at how mitochondria looks like. Mitochondria is a two-membrane organelle, so it has two membranes, an outer membrane and an inner membrane, and the inner membrane has many folds called cristae, C-R-I-S-T-A-E. It has many folds called cristae, and the importance of having all these folds will become visible in a few minutes. But between the cristae, there's a space inside where there's a material called the mitochondrial matrix. Like the cytoplasm outside, this is the material that is present inside the mitochondria. That's the matrix. Okay, so one of the key things that we are going to see here is how energy is held and transferred. Again, remember, the ultimate goal of cellular respiration is to produce ATP, adenosine triphosphate molecules. We have discussed this sufficiently in the previous chapters. Now, we want to make ATP, but we are not able to directly make ATP from the breakdown of carbohydrates. So we have intermediate high energy carriers. These are called NADH and FADH2. Remember, when we were studying photosynthesis, we looked at a molecule called NADPH. This is a very close relative of that, also derived from vitamin B, relatives of vitamin B. So NADH and FADH2. So the low energy version of this is the NAD+. NAD+ is able to take electrons, and remember, electrons, when electrons come in, very often you will have a H+, a proton also tagging along. So NADH is a high energy state where electrons along with protons are captured. So it's a charged, NADH is a charged molecule. Same thing with FAD+. FAD+ stores energy as FADH2. Okay, so now that NADH, the high energy molecule, can be broken down back into NAD+, and in the process you can release ATP. Um, and the same thing with FADH2 can be broken down back into FAD plus, and then in the process release make ATP. And how it happens is what we're going to see in the next few slides. All right, so let's break down this process. Um, so the entering molecule here is glucose. It starts with glucose and it happens in the cytoplasm, the cytosol. Glucose, a 6-carbon molecule, gets broken down into a 3-carbon molecule called pyruvate. The pyruvate wiggles its way into the cytoplasm, and in the cytoplasm it becomes a 2-carbon molecule called acetyl-CoA. That's called pyruvate oxidation. And acetyl-CoA joins the cycle where there is already a 4-carbon molecule present, and the whole objective of this cycle is to regenerate that 4-carbon molecule. And in the process, between pyruvate oxidation and the citric acid cycle, this cycle is called the citric acid cycle or the Krebs cycle. Between these two, you're producing a lot of carbon dioxide. Now, along the way, you are making ATP molecules, you are making NADH molecules, and you're making FADH2 molecules. And like we just saw here, NADH and FADH2 cannot directly be used for most for energy reactions, so it has to be broken down into a more convenient energy form called ATP, and that's the last step, which is oxidative phosphorylation, which happens as a part of the electron transport chain in chemiosmosis. So that's the electron transport chain. Glycolysis, you don't need to remember any of these long steps, but all you need to know is that it starts with glucose and it ends with pyruvate. Glucose is a 6-carbon molecule and it becomes 2 3-carbon molecules. Those 2 3-carbon molecules are called as pyruvates. Okay, this is the structure of pyruvate. Pay attention to this last COO group. That reminds you of carbon dioxide, right? COO. Okay, so just keep the idea of pyruvate in mind. You don't need to know the structure. Just know that glucose has become 2 pyruvate molecules. Now, to kickstart this reaction, you have to first add ATP. Right? You need to provide energy to start the reaction. So now you have a glucose molecule and you added ATP molecules, 2 ATP molecules, and you ended up with 2 ADP molecules. Okay, so that energy that was released by the breakdown of ATP runs this reaction, and this fructose, this intermediate molecule, what it does is it is broken down into another intermediate molecule and then broken down into pyruvate. But in that process, in this series of steps, what happens is NAD plus, so energy is produced at this point. Here you used energy, now you're producing energy. You're using 2 ADP molecules to produce 2 ATP molecules, and for this molecule again 2 ADP to 2 ATP molecules, and a net of 2 NADH molecules. So you're ending up with 2 NADH molecules and a net of 2 ATP molecules. When I say net of 2, because you have 2 ATP here, 2 ATP here, that makes 4 ATP, but you already used up 2 ATP. So at the end of the day, you're ending up with 2 ATP molecules from glycolysis along with 2 NADH. Now, acetyl-CoA, the pyruvate, the molecule that is formed here, enters into the mitochondria. There, the pyruvate, remember the COO that we were looking at earlier, that escapes as carbon dioxide. Okay, and the remaining molecule attaches to another molecule called coenzyme A and becomes acetyl-CoA. Acetyl-CoA. It's a 2-carbon molecule, acetyl-CoA. And in this process, we produce NADH molecule. Remember, you're creating 2 pyruvate molecules per glucose. So for one glucose molecule, the net NADH molecules here are 2 NADH. 2 NADH molecules are going to be produced along with acetyl-CoA, 2 molecules of acetyl-CoA. That leads us to the Krebs cycle. Now, Krebs cycle starts with oxaloacetate, this 4-carbon molecule called oxaloacetate. And remember this pyruvate broken down into acetyl-CoA. This acetyl-CoA comes and joins oxaloacetate. 2 carbons here and 4 carbons here. making a 6 carbon molecule. The coenzyme A is released, goes back into the pyruvate oxidation stage. So basically now you have acetyl CoA joined with acetyl group of this acetyl CoA joined with oxaloacetate to form citrate. Citrate is a 6 carbon molecule. Now the 6 carbon molecule citrate breaks down into a 5 carbon molecule. So that last carbon escapes as carbon dioxide. Now you have this 5 carbon molecule. Along the way, the energy that was released from that break is used to convert NAD plus into NADH. The 5 carbon molecule that is here loses one more carbon, the last carbon here, that goes off as carbon dioxide, and in the process it releases NAD plus becomes NADH. Okay, so and this 4 carbon molecule now kind of goes through a series of steps and converted back into oxaloacetate. Along the way, you saw NAD plus becoming NADH here, NAD plus becoming NADH, ADP becoming ATP, FAD plus becoming FADH2, and NAD plus becoming NADH again. So you're seeing a whole bunch of NADH molecules, FADH2 molecules being produced along with a little bit of ATP. So all the energy that is contained in the glucose molecule is now converted into the ATP molecules along with NADH molecules and FADH2 molecules. The electron transport chain is what takes the energy that is trapped in NADH2 and FADH2 and converts it into ATP. So as you will see, for every FADH2 molecule, 2 ATPs are produced. For every NADH molecule, 3 ATPs are produced. The process through which this whole energy production happens ends with chemiosmosis. So chemiosmosis is the production of ATP using the energy of hydrogen gradients across the membrane to phosphorylate ADP. It sounds like a mouthful, but let's take a look at it. This is not new to you. You remember ATP synthase we studied before in photosynthesis? That's what's happening here. You're going to use ATP. Nature does not like to reinvent things over and over. So it's reusing some of its old strategies. It's using ATP synthase, and the process of ATP synthase converting the rotor of ATP synthase moving and converting ADP to ATP, that is what chemiosmosis is about. And the rotor is initiated because of the difference in gradients. That's what this whole statement is saying. Alright, so remember, we are coming into electron transport chain with NADH molecules and FADH2 molecules. Now, the inner mitochondrial membrane has, as I mentioned, a lot of folds called cristae. The reason why it has a lot of folds is it's trying to maximize the amount of space that is where chemical reactions can happen, because this is the place, the inner mitochondrial membrane is the place where the electron transport chain is happening. So the more folds it has, the more electron transport chain reactions can happen, because all the material, all the protein complexes that are needed for electron transport chains are embedded in this place. So here in mitochondrial inner membrane, you have 4 complexes: complex 1, complex 2, complex 3, and complex 4. Okay. Now complex 1 is able to quick step back. NADH is going to break down into NAD plus 2 electrons plus H plus 2H plus. Okay. NADH breaks down into NAD plus protons plus electrons. The complex 1 helps in the breakdown of this NADH, and what it does is it takes in the electron. When it takes the electron, it opens up pores so that the proton that is created here is able to go through this. Okay. Now the protons are able to go from one side to the other side, and it's pumping it against the concentration gradient. So more and more and more protons are going to accumulate in this intermembrane space. Now FADH2, which was the other product of Krebs cycle, is broken down by in this step with complex 2. So FADH2 breaks down into FAD and FAD plus and H plus and electrons, and the electrons come and attach here, opening up space, once again sending all the protons from this side to the other side. And ultimately the electrons that are here, this high energy electrons, are transferred to 3 and then find complex 3 and then finally to complex 4. In complex 4, inside this complex, you need, so at this point again you have electrons here, but you need something to accept the electrons because it cannot keep on moving it, passing on the electrons. These guys were happy to just pass it on to 3, and then 3 was happy to pass it on to 4, but 4 says the buck stops with me, you know, so I need to do something about the electron that I'm carrying. That's where the breakdown of water becomes a big deal. Water, I'm sorry, the breakdown, please ignore the last part. But this is where accepting of the electron becomes important. So what happens here is that you see, when the electron binds here, protons start moving from here, right, from here to here. The oxygen that we breathe in, is able to bind to the electron that was transferred to complex 4. So the oxygen that we have breathed in binds to the electrons, and the protons that are passing through this from here to here are able to bind to that oxygen with the electrons. So protons that are moving along with the oxygen that we breathed in, along with the electron that was transferred from here, and the electrons were transferred because of the energy of NADH and FADH2, and those electrons, so together come join and form water. Okay, water is formed. So the final electron acceptor in this case is oxygen. Oxygen is the final electron acceptor. But see what has happened on this side. You have created a whole lot of protons in this intermembrane space between these two membranes, and that has created a gradient. Remember our definition of chemiosmosis. You have you have H plus gradients created, and that H plus gradients, there's more here and less here, causes the activation of ATP synthase. ATP synthase is going to allow the movement from facilitated diffusion of protons from one side to the other side. And ATP synthase is this beautiful molecule which has got this really nice rotor at the bottom. So as protons move from here to here, that rotor gets activated, and ADP molecules become high energy ATP molecules. So for every NADH molecule, you can create enough protons to create 3 ATP molecules. For every FADH2 molecule, you can create enough protons to create 2 ATP molecules. So we came into this reaction from Krebs cycle with a total of 6 NADH molecules and 2 FADH2 molecules. Okay, 6 NADH and 2 FADH2 molecules. Remember, if 6 NADH molecules are present, then you have 3 ATPs per NADH, so 6 times 3 is 18. For every FADH2 molecules, 2 ATP molecules are produced, so 2 times 2 is 4. So 18, 6 times 3, 18, plus 2 times 2, 4. So we have 22 molecules of ATP just created in this process. Okay, 22 molecules created just from NADH and FADH2. Plus also, remember, you have NADH coming from these two steps. You have 2 NADH2 coming from this step, and you have 2 NADH coming from this step. So 2 times, so 2 here, so 2 times 3 will be 6, 2 times, so 3 ATP molecules per NADH, right? So 2 NADH from here, so 2 times 3 is 6, So add that to the 22 that we created just from the Krebs cycle. So we have 28, and then we have 2 NADH here, so 2 times 3 ATP molecules. So we have 6 ATP molecules here, so that is 6 there. So 28 plus 6, that is 34 ATP molecules so far. Let me see if I can show you a reaction here. So 2 ATP molecules were produced in glycolysis along with 2 NADH. That 2 NADH becomes 6 ATP molecules, and in the pyruvate oxidation we produce 2 NADH, that's equivalent to 6 ATP molecules. In the Krebs cycle we created 2 ATP molecules plus 6 FADH, which became the 18 ATP molecules, and 2 FADH2, which became 4 ATP molecules. Together, you add it all together, you end up with 38 ATP molecules. This is in an ideal state. It's not always 38. Sometimes you have less, sometimes you have even less. So 38 is the maximum amount of ATP molecules you can produce per glucose molecule. Sometimes people estimate it at 32 because they say there's no NADH produced in this step, but ideally this is what you might expect. So the key thing here is this reaction will not proceed if there is no oxygen to receive the final electron here. If there was no final electron, this reaction comes to a standstill. So cellular respiration will not proceed. But remember, in the early days of Earth, there was no free oxygen available. Okay, so what happened in those times? So there was a different type of chemistry involved there, where instead of instead of oxygen as a final electron acceptor, we had other chemicals like sodium sulfate or methane or nitrates, sodium nitrate. All of these were used as final electron acceptors. So when we are so these are reactions that can proceed in the absence of oxygen, and this is not very energy efficient, but that's a way of survival in the absence of oxygen. In fact, those organisms still exist today as methanogens and other archaebacteria that live in sulfur containing conditions and so on. This is called anaerobic respiration. But sometimes you don't have to go through this whole. Some organisms have a strategy where they don't have to go through this whole process in the mitochondria. If you think of a single cell organism like yeast, in the absence of oxygen, it can completely bypass all of these steps, and it can just go from glycolysis, which is pyruvate formation, to creating alcohol and carbon dioxide. That is called as alcohol fermentation. Or it can, or in our bodies and in some organisms, some bacteria can do this, where pyruvate in glycolysis is converted into lactic acid. Again, remember that none of these processes are energy efficient. But it helps the organism survive as a strategy in the absence of oxygen. Okay, so so we have anaerobic respiration, and then we have lactic acid fermentation, and we have alcohol fermentation. Let's take a quick look at lactic acid fermentation. So glycolysis happening in the cytosol. Remember, glycolysis does not require oxygen anyway. All it is doing is converting glucose into pyruvate in the series of steps we saw before. And in the process, we have a total of 2 ATP molecules being produced and 2 NADH molecules being produced. But if the NADH is not going to go into the mitochondria, and remember, one of the byproducts of mitochondrial of the electron transport chain is NAD plus and FAD plus. So if there is no NAD plus being produced here, it means all the NAD plus is going to be used to make NADH. So you have a whole bunch of high energy NADH molecules and no way to use that energy. So we need to regenerate the NAD+. So that happens when pyruvate becomes lactate. When pyruvate becomes a molecule called lactate in the absence of oxygen, and lactate becomes lactic acid, what happens is NADH becomes converted into NAD+. So now the NAD+ is available for further conversion of glucose into pyruvate. So the whole objective of lactic acid fermentation is to regenerate NAD+, so that glucose to pyruvate conversion can happen, and in the process you can get a small amount of ATP being produced. Same thing with alcoholic fermentation. So glucose becomes pyruvate and NADH is produced, but the NADH needs to be converted back into NAD+. So the pyruvate, instead of becoming lactic acid, the strategy here is to release the last carbon here as carbon dioxide. Okay, the last carbon, we saw pyruvate has a COON, right? So the last carbon is going to be last as carbon dioxide, and the remaining molecule is called acetaldehyde, and the acetaldehyde becomes ethanol. Ethanol is alcohol that is, it's usually consumed in alcohol in alcoholic beverages and things like that. So you're producing carbon dioxide and you're also producing ethanol. But in the process of conversion of acetaldehyde to ethanol, you're converting NADH into NAD+. So now you have enough NAD+ to keep running the glucose to pyruvate reaction so that you can produce this meager amount of ATP. Okay, so you see in bread, when you're making bread, you are able to make, you're able to convert, you're able to see the rising of bread, right, which is the production of spaces within the bread. That is all carbon dioxide and a little bit of ethanol that is produced, and some of the organisms can be used to make beer and fermentation for alcohol, like wines and things like that. Okay, so that is cellular respiration, and you see how beautifully it all comes together. Life starts, or the energy play starts with photosynthesis. Energy from the sun gets into the light dependent reactions inside plants, producing NADPH and ATP, which are then used in the Calvin cycle, or the light independent reactions, and at the end of that you are producing glucose, and alongside when the light dependent reactions are producing oxygen. Now when we need to use this glucose, the glucose is used along with oxygen from the atmosphere. So the glucose is used along with the oxygen that was produced by the plants in in this step for glycolysis, producing so the oxygen comes in later, but the glucose is used in glycolysis to make pyruvates, and pyruvates are then broken down into acetyl-CoA molecules, and they enter the Krebs cycle, and the NADH and the FADH2 that are produced here are converted into ATP molecules as part of the electron transport chain. So you end up with a ton of ATP, and in the process oxygen comes in and carbon dioxide is released. So now the carbon dioxide is once again a source for the light independent reaction. And so this whole system is dependent on each other. Photosynthesis and cellular respiration depends on each other, and ultimately we are able to take the energy of the sun and capture it into ATP molecules. So you can see why we need lots of trees in our environment, and why we need animals, and how a balance in numbers between animals and plants are needed, and what could happen, again, as a big picture thinking. Think about what would happen when we cut down trees in enormous quantities, and you are compromising this part. What would happen to the amount of oxygen that is produced, and what would happen to the amount of glucose that is produced in our food chains and food webs. Okay, hopefully this all comes together for you, and by now you should be able to describe cellular respiration, what its objective is, and what the three steps of cellular respiration and that intermediate process of pyruvate oxidation, and how energy flows across the cellular respiration process, and how anaerobic respiration and fermentation can happen in the absence of oxygen as a final electron acceptor. Okay, hopefully this was helpful. Thank you.